let's start this. Okay, so hello and welcome to Excelsior Academy. We are starting here with cell biology, and the first topic what we are going to discuss is the biological membrane. So basically, we know what plasma membrane is. So in this topic, slowly, we'll talk about the discovery of the plasma membrane, how plasma membrane is arranged, what are the different types of the phospholipids present, and their function also. Okay. So biological membranes, we'll be talking about different types. So it is the fundamental structure of cellular evolution. Now, why do we say that it is the cellular evolution over here? Akshaya, can you put some thoughts over here? Like why the plasma membrane is known as the fundamental structure of cellular evolution?
Ma'am, because mitochondria was present and then it was engulfed by some other cell. So that's how, and I guess so. So to keep everything into place, cellular membrane came into function.
No. You need to go beyond that. The first cell which was formed, it was all because of the plasma membrane. If you remember, see what you talked about is the endosymbiotic theory. Okay, jahan pe the first mitochondria, the first chloroplast came because of the engulfment of the blue green algae, and the mitochondria, the other bacteria eating another bacteria. Okay, but here it is the fundamental structure of cellular evolution, we say, because when, if you remember, in the case of the evolution, you must have talked about or heard about Oparin and Haldane
Yes, ma'am.
tube and the primitive atmosphere. Then it has rained for several million of years, so that all the mountains were getting, eroded off, and all those nutrients came into the ocean, and that's how our earth has got the 75% of the ocean water, correct? Now the thing is that the first life has evolved in the water, we all know. Now how it has evolved, it is because all these phospholipids kind of thing, they start coming together because they are hydrophobic in nature, right? So they try to come together. So when they're coming together, they started getting circularized like this. Okay. And this resulted in the formation of your first cell. So pehla plasma membrane aapka bana because of the accumulation of the phospholipid. And when this accumulation, this aggregation was happening, that time certain amino acids, certain carbohydrates got integrated inside. And that resulted in the formation of the first enzyme or the first carbohydrate required by the organism. So this is how your first cell was evolved. And hence it is known as the fundamental structure of the cellular evolution. Okay. अभी थोड़ा सा clear हुआ? Don't worry, because this is a part of evolution. I'll be taking there in detail. Okay. Now, okay. So now we know that these prokaryotes and eukaryotes are surrounded by plasma membrane. Okay, so now we know, as I said, prokaryotes and eukaryotes are surrounded by plasma membrane. So this is important, that it is going to define the cell boundary, separating its internal contents from its surroundings. So this we know, right? In the case of the eukaryotes, you have got cytoplasm, then the nucleus is there, right? The nucleus has got its own components, and the cytoplasm has got its own components. So mitochondria हो गया, फिर chloroplast हो गया, ये सब के अपने अपने components हैं, and everything is defined by the plasma membrane. Okay, and moreover, it is allowing the selective passage to molecules, hence determining the composition of cytoplasm. So we know कोई भी चीज easily pass नहीं कर सकती plasma membrane को. It needs to have certain characteristics. Now, what are those? Like first, it should be permeable, right? Through the plasma membrane. तो यानी कि उसको होगा rich in lipid content होना चाहिए, right? Small in size होना चाहिए, so that the bulky carrier or the bulky molecules cannot pass. It becomes difficult for them. Okay, so we'll talk about all those parameters which allows any molecule to pass through the plasma membrane. Now let's talk about the plasma membrane. So it is made up of phospholipids as well as the proteins. Okay. So guys, these are like very basic. आपने पहले से ही पढ़ा होगा. We are just revising here. So phospholipid क्या है? It is impermeable to water soluble molecules. But if you have heard of it, we say that oxygen can pass. Okay. And then देयर आर सर्टेन मॉलिक्यूल लाइक कोलेस्ट्रॉल हो गया, आपका ये सब पास कर जाएगा, ठीक है. सो व्हाई कोलेस्ट्रॉल कैन पास? बिकॉज ऑफ इट्स हाइड्रोफोबिक नेचर. नाउ व्हाई ऑक्सीजन कैन पास? कैन एनीवन टेल मी? So it does not need any carrier or any channel by which it needs to get passed through the plasma membrane, but it can easily pass through the plasma membrane. Why? Yes, it is because of its small size. Okay, so it is easy for oxygen, for water molecules to pass through the plasma membrane because the size is very less. Now coming to the proteins. So proteins are responsible for the passage of the ions in organic molecules. It also helps in mediating interactions between cell and its environment. So already हमें पता है plasma membrane में we have got different types of proteins, correct? If you remember, we have got peripheral proteins and we also have integral proteins. जिसमें से integral proteins are responsible for your cell to cell interactions, जो हम एंटीजन एंटीबॉडीज इंटरेक्शन बोलते हैं, या सेल टू सेल लाइक फॉर द मूवमेंट ऑफ मसल और द मूवमेंट ऑफ द मैक्रोफेजेस inside the blood vessel. So all these are responsible or the duty of the integral proteins. ठीक है. Now when we talk about that, this is suppose the plasma membrane, outer and inner, कोई protein है आपका ऐसे, okay, and it is allowing the passage of, let us say, acetylcholine. So we know that it's a ligand for your receptor during the synapse, right? So यहाँ पे आपका क्या है? आपका एक channel है which is allowing this passage of the acetylcholine. So even this channel is made up of proteins. ठीक है? So all this we are going to discuss in detail with the structure and the function. Now more coming back to the previous, that is about the phospholipids. So phospholipid mein you have got the polar head and the non-polar region. So even if the molecule is polar in nature, it can interact with the polar head of the plasma membrane here only, but it is difficult for it to cross. Okay? Similarly, if you have got any lipid molecule, if it is bulkier, okay, then it is easy for it to pass. But agar chota sa lipid molecule, and then this polar will not allow that lipid molecule to pass through. Okay? तो हमें ये भी देखना होता है कि how much hydrophobicity or philicity is present. Okay. Now coming to the composition. So as already mentioned, they are made up of phospholipids and protein, and the fundamental structure of the membrane is the lipid bilayer. Now अगर आपसे कोई पूछे कैसे पता चला हमें plasma membrane के बारे में, the simple answer is RBC. Now guys, I'm asking you, why the scientists have taken RBCs to study about the plasma membrane? RBC में ऐसी क्या खासियत थी that they were happily using RBCs rather than any other eukaryotic cell? Yes, anyone can tell me the answer? Yes, Akshaya?
The nucleus is not there, they're enucleated, so it is very easy.
Okay, yes, the correct answer is yes. The nucleus is not present. So it's not only the nucleus, it is also the other organelles, they are present in are absent in the RBCs. So kaise hoga? So when they have to harvest the plasma membrane, it was easy because there were less interfering components of the cell. Okay, so it was easy for them to harvest the plasma membrane. So when they harvested and they found that it has got the phospholipid bilayer and the proteins embedded. Okay. Now, so these were the scientists. This is just for your reference only. So in 1925, this Dutch scientists by the name Evert Gauter and the Francois Grendel, they extracted the membrane lipids from a known number of RBC corresponding to a known surface area of plasma membrane. Okay. So why it was necessary to have known number of sorry. So known number of RBCs. See, ये आप कैसे calculate करोगे exact number of RBC? There is this experiment by hemocytometer, आपने सुना होगा, correct? Which has got chamber like this. जहाँ पे ये 25 रहते हैं, you have to calculate. यहाँ पे आपके 20 रहते हैं. Have you seen and used?
Yes, ma'am.
Yeah. So
Yes.
depending on that, you're going to get the total number of RBCs used for the experiment. Okay. Now, it is just approximated that one RBC might have this much of surface area because you know RBC का इतना area है, इतना surface area होगा. Okay. Let us consider that this is spherical in shape. Now, अगर हमें मालूम है that 5000 RBCs है, तो उसका surface area will be that much multiplied. So now they have got this value. Okay. What they have done is they have extracted the layer of mono layer of the lipid. Okay. So आपका RBC ऐसे है, तो उन्होंने ये extract किया. Okay. And then they studied. What they found was that every time when they are doing the experiment, they are getting twice the reading. अगर एक RBC में मैं इतनी काम कर रही हूँ and I'm saying that one RBC should correspond to this much of surface area, but fortunately, unfortunately, these people were getting twice the number. Okay. Later, with the help of the electron microscope, it was found that because since the layer, lipid layer is bi, okay, there are two layers. That's why every time they were getting twice the surface area. Okay, and then it was concluded that the plasma membrane is not a monolayer but a bilayer. अब हम क्या है, हम जब से पढ़ते आ रहे हैं हमें पता है that plasma membrane is bilayer, but these were the people who found out that it is a bilayer. So we always need proof, evidence. Okay, so this is the electron micrograph of a plasma membrane, and you can see that this is the air and the water interface. ठीक है, यहाँ पे आपको दो heavy ऐसे line दिख रहे होंगे. So that dark stain, okay, it is because the polar head was stained with the heavy metal, and the inside one, जो आपका phospholipid layer है. See if I draw it like this. So this polar head like this, so they were stained heavily, and inside region, the hydrophobic region was stained light, and that's why you can see two lines are going like this. Okay, hence it was concluded that the plasma membrane is made up of bilayer. Okay, clear?
Yes, ma'am.
Okay, now let's talk about plasma membrane components. Anyways, this in detail we have to do. So what we need to understand over here is that each plasma membrane, they have got the proteins present, correct? Already discussed. So we have got the peripheral proteins, we have got the integral proteins. As I mentioned, integral proteins का क्या काम है? They are involved in the channels, they're involved in the interactions. Now, what about the peripheral proteins? So peripheral proteins, they are present over here. As you can see it here, these ones. Okay, so they are present over here. इनका क्या काम है? To understand the reaction stimulus taken from here, give it to over here, and then inside reaction media. ठीक है? So they are just passing the signal from outside to inside. Then you have got the cholesterol also embedded in the plasma membrane layer. Now, Tanvi, can you tell me what is the role of cholesterol in the plasma membrane? Yes, Tanvi? Would you like to try?
Does it contribute to the hydrophobicity?
Okay. But why do you want to make plasma membrane more hydrophobic?
It is better protected because the more hydrophobic it is, the less the chances are of outside components coming in.
What if your outside components are hydrophobic in nature itself? Even your toxins are made up of steroids, so they can easily pass through plasma membrane, easily can destroy the cell.
Actually, since it's just the tails, right? In the tails, cholesterol gets in, right? It'll probably be the interference other than the tails that is required to stop molecules from coming in, maybe.
No. So that is not the correct answer. So cholesterol is important for each organism. ठीक है? So right now अभी scenario कैसा है? कभी हम बोलते हैं ना कि अरे आपके में cholesterol ज़्यादा है. So what does it mean that time? That bad cholesterol is more in your body, which is getting deposited in the blood vessels. ठीक है? जिसकी वजह से the passage of blood is becoming very narrow. Correct? Now when we talk about the presence of cholesterol in the plasma membrane, हम लोग एक cellular level तक जा रहे हैं. So there its function is to maintain the fluidity of the plasma membrane. Fluidity of the plasma membrane. So what does it mean? It says that your cell shouldn't be so rigid, otherwise the movement cannot happen, and it shouldn't be like water कि वो कुछ भी contain ना कर पाए components को. ठीक है? So it has to be in between, a gel-like composition. So if your cell is present at high temperature, let us say that your particular cell is present at high temperature, so that time the cholesterol should be less, because more the cholesterol, more the fluid-like property of the plasma membrane. ओके, सो हाई टेम्परेचर में मुझे कोलेस्ट्रॉल कम चाहिए, नहीं तो क्या होगा, मेरा जो सेल है, द प्लाज्मा मेम्ब्रेन है, वो एकदम पानी जैसा हो जाएगा. सो व्हाट विल हैपन इफ इट बिकम्स मोर फ्लूइड? It is easy for the cell then to lose all the components, right? वो drain off हो जाएगा. Now just imagine the another scenario. You have kept your cell at a very low temperature. Let us say that आपने फ्रिज में रख दिया या फ्रीजर में रख दिया. Okay, that time I need cholesterol to be more. Why? Because at low temperature the cell is going to freeze. It is going to become rigid in structure, which can hamper or destroy the cell. Okay, I don't want that to happen. So the organism will understand and make more cholesterol to get inserted into the cell, so that it will have more fluid-like conditions. तो आपका cell normal condition में होगा. ठीक है? समझा, श्रुति?
Yes, ma'am, but I'd also heard that the temperature change causes things to go to the fatty chains of the bilipid itself. And the bilipid itself is usually fluid, but then temperature affects it a lot.
So sorry for the disturbance. Yes, Shruti, what you were saying?
I heard that the lipid membrane itself is usually fluid enough, because when there is a difference in temperature, it causes kinks to happen in the fatty acid chains, and that is what affects fluidity.
Yes, yes. So there we are going to talk about the saturated fatty acid and unsaturated fatty acid, right? So even this is going to affect the same fluidity of the plasma membrane. So we'll talk about that. So cholesterol is the add-on. मेन काम तो आपका फॉस्फोलिपिड सैचुरेटेड अनसैचुरेटेड ही करते हैं. सो मोर सैचुरेटेड मींस मोर रिजिड स्ट्रक्चर. इफ यू हैव अनसैचुरेटेड, मोर किंक विल बी प्रेजेंट, सो हेंस मोर फ्लूइड लाइक नेचर. करेक्ट?
Yes, yes. Yeah.
Right, this is what we have studied, right? But in addition to that, organism tries to keep the cholesterol also in between to increase the fluidity of the plasma membrane. Now this is one factor. Second, I don't know whether you have heard or not, there's something called lipid raft. सुना है क्या कभी? Ever heard about it? नहीं. See, raft means what? Everyone knows, right? किसी भी नदी में या कहीं पे भी एक टाइप का फ्लैट बोट जैसा कुछ रहता है. Okay, so you can just take this raft anywhere in the surface or in the lake or in the river. Same way, plasma membrane, since they are not rigid, they are mobile. Okay, this particular phospholipid, if it is present right now here, after some time you will see it is present over here. So each phospholipid is mobile in nature, and that's why it is always in the dynamic nature. Okay. Now this cholesterol along with your saturated and unsaturated depending on the requirement plus sphingomyelins, they come together and form lipid rafts. अब इसका क्या काम है? Because they carry the most important integral proteins which is required for the interactions. कभी आपने सोचा कि कोई सेल है, okay, उसको बैक्टीरिया इन्फेक्ट कर गया है, so that particular cell decide to undergo apoptosis or getting killed by the our natural killer cells or the macrophages because the cell has got infection. अगर वो अपने आप को burst करेगा, so what will happen? The bacteria will spread and the other cells will get infected. Correct? So the cell decides to undergo the apoptosis or the death by the natural killer cells or the macrophages. Now ये चीज कैसे वो समझाएगा macrophages को? By bringing all the lipid rafts together, which carries those integral proteins as the interactions, they give the signal to the macrophages to come and eat them. आया समझ में? It's all getting mixed right now. Cell bioimmunology थोड़ा सब mix हो गया है, but point is clear. Anyways, we'll be talking about lipid raft also in detail. Are you getting the point here? Like क्या convey करना चाहती हूँ मैं? हाँ, थोड़ा बहुत. Okay, maybe just forget it. We'll come to that portion slowly, because this is the entire cell. See, that's what I said. Huh? Once you complete all this thing, you can then interlink. Right now, I just talked about all cell biology and immunology mix. Correct? It's okay. Just leave. Let's go with the simple basic things. Plasma membrane, आपके proteins रहते हैं. Integral and the peripheral proteins. You have got the saturated, unsaturated. Here also we'll talk about each, right? And one more thing is that they're also having the glycoproteins and the glycolipids. Simple. They have got the carbohydrate moiety. Okay. Now, to understand the plasma membrane structure, you need to know the technique known as freeze fracture electron microscopy. So I'll be taking this in separate lecture. So just remember what is this freeze fracture electron microscopy. So we'll be discussing about this. Now let's talk about the role of the carbohydrates and proteins. So they are mainly to help in the cell-to-cell recognition. जो हम interactions की बात कर रहे थे. ठीक है? I'll just try to give one small example like how carbohydrates and these integral proteins are helping. So have you guys studied in immunology or anywhere when a two cell interacts? Let us say that this is your macrophages and this is your some cell. So what happens when these two are going to interact? Okay. अच्छा ये भी छोड़ो और सिंपल तरीका मैं देती हूँ. RBCs are there. Okay, so RBCs are there. We know that the grouping is present in RBC, our blood group system. So A is there, B is there, AB is, and O is there. Now, since you are from animal physiology background, you must be knowing what this A, B, and O stands for. Correct? Yes, Akshaya?
Yes, ma'am.
Right. So you know why it is A, why it is B. It is because of the, yes. Would you like to give the answer, Akshaya?
Protein present on it.
No, it's not the protein. It's not the protein. See, that's what I'm coming to the point. It is the carbohydrate. If you remember, it has got the glucose, right? So we'll come to that. It has got the N-acetylglucosamine. What are these? These are carbohydrate, sugar moieties, right? Which is present on the proteins. Integral proteins की यहाँ पे वो carbohydrate present है ऐसे. Okay. Now, which does not have these as antigens A and B, then it is your O. When the cell is having both of these kinds of sugar, then it is AB. Correct? So these carbohydrates will be recognized by the antibodies. Antibodies के पास क्या है? वहाँ पे protein. So the protein carbohydrate interactions will give a signal to your system that wrong blood or something is wrong with the grouping system or the antibodies are present. So it is going to react, and that's how your immune system is going to behave. Okay. So that's why the carbohydrates are present. So they are present in small quantity or less percentage, but they are the main player in the cell to cell recognition. Okay. Is this much clear? Okay. Now, proteins, they will be forming the pumps and receptors. And believe me, guys, this cell bio first topic, we are going to talk about different types of receptors and all. So much is there, like ABC, ABC cassette is there, where then we are going to talk about the different types of pumps like V pump, F pump, P pump. Okay, then different channels, ligand gated, voltage gated, mechanoreceptors. All these will be covered just to understand how different proteins present on plasma membrane behave and how eventually it is helping in the cell to cell interactions. Okay, now lipid composition, again this is very simple but very important to remember. So we have got PC, PE, PS, PI, phosphatidylcholine, ethanol, amine, serine, inositol and sphingomyelin. इसमें से भी आपको क्या मेनली याद रखना है अबाउट दी PS, phosphatidylserine. ठीक है, सिंपल है, आपको बस इतना याद रखना है that serine, inositol and ethanol amine is present on the inner leaflet of the plasma membrane. Okay, and rest all is present on the outer membrane. Why it is important is that because when there is a apoptotic signal given by the cell, okay, how does the macrophages understand that this particular cell is undergoing apoptosis? अब वो cell तो मर गया है, but वो debris present है inside the system, right? It needs to be cleared off, correct? वो डेड बॉडी हमें हटानी है. वो डेड बॉडी कैसे हटाएगा? जब तक आप उसको सिग्नल नहीं दोगे. वो कौन करेगा? मैक्रोफेजेस करेंगे. तो वो सिग्नल आप कैसे दोगे मैक्रोफेजेस को कि हाँ बाबा ये डेड बॉडी इसको हटाना है, राइट? So that signal is provided by the phosphatidylserine. So when apoptosis happens, जो phosphatidylserine है जो आपका inner leaflet में present है, okay, always present on the inner leaflet. But if the particular cell is undergoing apoptosis, PS will go to your outer leaflet. And this is the signal which will be recognized as apoptotic signal by the macrophages. Clear? Please always remember about this. ये बहुत बार आया है exam में. Okay. Yeah, same thing which I just described now. So apoptosis signal by the PKS translocation. So क्या होगा? So we have got the different enzymes are there. I'll ask in the next lecture. Like हमारे पास है flippase, we have floppase and scramblase. ठीक है? तीनों का एक ही काम है. But still all three have got the difference. So what is the difference? I need to know from you guys. So 3 enzymes hai, you are 3 people, so tomorrow you are going to answer. Is this assignment clear to you? Today's assignment?
Yes, ma'am.
Okay. Yeah. Now coming to the functions of the phospholipids. So I already mentioned they are impermeable because of the hydrophobic fatty acids. So it does not allow anything to just cross. And fluid-like, so it can move freely in the interior of the membrane. So this fluidity of the cell membrane is very important, and how inside the cytoskeleton structure which is present. As you know, cytoskeleton means it is made up of microtubules, intermediate filaments, actin filaments, right? So actin filaments plus the fluidity of the cell will make your cell move from one place to another. एक example देती हूँ. I hope you guys can imagine then. Let us say that this is your blood vessels. Blood vessels are made up of endothelial cells, if you remember. Okay, they are made up of endothelial cells, vascular vessels. Now you have got macrophages coming. Okay, macrophages कैसे काम करता है, या कोई भी WBC अगर है या any other defensive cells are there, so how they are working. Let us say that you have got wound over here. आपको चोट लगी है यहाँ पे. So you know that the inflammation will be there, leakage of the blood will be there. So we need platelets, right? So पहला काम क्या होता है? Platelets लाके जो blood loss है उसको बंद करना. Inflammation होगा because क्यों? Bacterial exposure हो गया है. We need to get more WBC so that they can attack all those bacteria. Macrophages is also required. Now let us say that I'm talking about the macrophages over here. Now if my cell was very rigid, ऐसा का ऐसे ही रहेगा. So it is not possible for them to go between the two endothelial cells. ठीक है? आपके blood vessels में endothelial cells के बीच में gap नहीं होता है. Gap होगा तो क्या होगा? हमारा blood loss होगा, correct? So it is not a good capillary. It is not a good blood vessels. But it should be in such a way that if required, this macrophages should come, slow down, it should come over here and squeeze itself from here and reach the wound area. ठीक है? So exactly अभी क्या हुआ यहाँ पे? So the protein-protein interactions between the two endothelial cells was broken and made new interactions with the macrophages. So you can see that macrophages ने कैसे अपने आप को पूरा squeeze किया, correct? Now this is only possible because of the fluid-like property of the plasma membrane. Okay, and the interactions which they make with this particular cell ऐसे and ऐसे. So the interactions which were broken between these two are made new interactions with the macrophages and again breaking them and moving towards the wound site. तो क्या हुआ eventually? ये cell ऐसे से move हुआ, यहाँ पे आया, यहाँ पहुंचा. Okay, is this much clear? Now this is only possible because our plasma membrane has got the fluid-like property. Rigid होता तो वो cross नहीं कर पाता. पानी जैसा होता तो कहीं पे भी फैल जाता. So the inside the macrophages which it has got the property of killing the bacteria and all, it won't be able to radiate. Correct? Yeah. Any doubts over here? Clear? Yes, Tanvi, is it clear?
Yes, ma'am.
Okay. Now the factors which is affecting the membrane fluidity. So as already mentioned, we have already discussed this thing, that is about the unsaturated fatty acids and the saturated. Okay. Now let's talk about this in detail. So we know more saturated means more rigidity because they are having the straight chain. तो जितना ज्यादा straight रहेगा उतना आपका rigid packing होगा. Correct? Shruti? Correct?
Yes, ma'am.
Now, if you have got unsaturated, you are going to have the kink, because of which the packing is not going to be rigid. It is going to be loose packing, and that's why more fluidity is there. Now, let's talk about the cholesterol. So, how cholesterol is doing? So, it is decreasing the mobility of the phospholipid fatty acid chains. So, kaisa hoga? See, I don't know how to explain this because here lipid raft is also coming. Just imagine that this is your upper layer, okay, upper layer of the phospholipid. As I already said that एक particular phospholipid अगर यहाँ पे है, थोड़ी देर बाद वो यहाँ पे भी होगा, correct? ये आपका फिर दूसरे chain में चला जाएगा, like from outer to inner also it can go because of the help of those enzymes like lipase, floppies and all. Is this much is clear? That any phospholipid can move from here to here, here to here. Okay. Now I don't want all of my phospholipids to move like this. नहीं तो मेरा cell कभी stable हो ही नहीं पाएगा, right? The integral proteins it is holding will be lost then. वो कुछ भी float करेगा, जैसे पानी में कोई चीज आप डाल दो वो float कर रहा है तो क्या होगा, वो float करते करते कहीं भी पहुंच जाएगा, right? But I don't want that thing to happen. I want that it should be in control. Let them float, but it should be in control. So whenever any interaction is required with the another cell or any bacteria or whatever, it should be present over there, ready to get interacted. ठीक है? Cholesterol आपका fluidity भी increase करेगा, plus it is going to restrict its mobility. अब clear है थोड़ा सा? And we'll talk about lipid raft in detail in the next upcoming lectures. Okay? I'll show the figure and the diagrams and the animations are there. So अभी तक के लिए clear